Good morning, everyone. Welcome to today's session. And Dr. Kamau, are you there? Hello? You can start the recording from that side. I can't start it from here. Hello, so we are going to start the training. So this morning, again, we are privileged to have Dr. Paul Kachaplula to take us through the session. So we are starting at 10:30, then it will be a 2-hour session. We hope he's going to break at half when he has done 1 hour to allow just a few questions, then we continue. So today is the last day of the training. We're going to have the last 30 minutes as a valedictory session. So you may go ahead, Dr. Paul, and start the training.
Thank you very much, Dr. Mfungwe. I noticed you were trying to contact Dr. Kamau for the recording. Have you managed? Is he recording?
No, he's not responding. And I can't see him on the participants list here. Let's see. I don't know if I can't see his name here.
I noticed there is a Sonam is a host from AARDO. I don't
know
who else is host.
Okay, yeah. So Sona, I don't know. Sona, if you can get us. Oh, I can reclaim the hostship, I think. Let me see.
Okay, try to do that, we see. And then you can make many more as co-hosts.
Oh, yeah. Okay, so we can start the training. So the session is being taken by Dr. Paul Kachaplula. So you can run us through the session, Dr. Paul, it's your turn.
Thank you very much, Dr. Mfungwe, and thank you participants once again for meeting in this manner to share a few ideas. It's really an honor to present to you. You are experts and professionals in different fields and from different countries. It's such a pleasure to be addressing you. So we have over the past 3 days, gone through several modules, as you recall, for those who've been with us throughout the 3 days. But for those who have missed some of the days, what we did was on the first day, to begin with, this is in general, the whole training is attempting to look at how to harness modern technologies in dealing with plant diseases and pests. That's both insects and weeds. And we broke that into 4 modules. The first one, which was on day 1, was modern concepts in insect pest management. The second day, we looked at the role of plant breeding in this issue of dealing with plant pests and diseases. And yesterday, we looked through modern concepts as they relate to the management of plant diseases. And like I said, today we are focusing on weeds and nematodes. As a way of starting, we know that weeds are a real problem in agriculture. Globally, about $100 million is lost in crop production as a result of weeds. And it's much worse in developing countries, including a lot of the AARDO countries that are in the developing world. where you can have up to 30% losses in crop productivity because of weeds. And the 30% is actually a conservative estimate, because in many places, especially if you pick a specific crop, you find that there's crops where unfortunately our farmers are experiencing even more than 50% losses as a result of weeds. So the image you see on your far left is an example of a field, what could happen in a field due to struggles in weed management. And of course, it's not that our farmers love to see a scenario like this. No, there is several unique problems associated with weed management. And one of them is that if you're trying to use some of the conventional methods, weeding, for example, hand weeding is one of the things that a lot of our small-scale farmers in many other countries in Asia and Africa use. So that requires a lot of labor. So sometimes a farmer might, plant a certain field, certain size, anticipating that members of the extended family are going to be available and they will help in the weeding. But sometimes, due to seasonal variations and unpredictability, sometimes in availability of family labor, you end up with a situation like this. And obviously, this crop here, this farmer is very lucky if they're even going to see a single cob out of a field like this. Then the image in the middle is this could be a sorghum plant, or it could be a maize plant, or it could be a cereal such as rice or wheat. Okay, that big stem. And then this pretty flower you see around here, it's not for decorations, and it's not as innocent as it looks. It looks so pretty and nice, but it's not as innocent as it looks, and I'll explain to you shortly why. And water bodies, you find rivers and lakes, sometimes they get clogged up by weeds in this manner, and the utility obviously of a water body like this becomes problematic if you want to, if it's near, so you want to use the place for your paddy rice or fishing, or even just electricity generation. A clogged river system like this becomes a big problem, and this is what happens because of weeds. Now, the costs of pesticides, in this case specifically herbicides, it's really very high. For you to effectively invest in these herbicides and continue being productive, it really costs a lot. All these add up to the $100 billion and even more in some cases. Now, how do the weeds actually cause losses? Let's look at this a little bit close. And I realize a lot of you professionals in here, you know a lot of how these processes occur. But let's just run through them before, so that we're on the same page, even as we build on this story to get to modern management. So a crop like this, as you can imagine, there's a lot of inputs that goes into here. You may have applied some type of fertilizer. You are obviously irrigating this crop, and there's sunlight needed for the plants and so on. These weeds that are in the middle of these rows, they are going for those same exact resources. They are going for your water that you're giving your crop. If you have added some organic or inorganic fertilizer, they are going for the same nutrients. And in the end, you have this competition for growth resources. So in the end, you do not have, your crop does not have adequate amounts of that resource. And in the end, depending on the complexity or the extent of the weed infestation, you may not even harvest anything out of here. And this is a sorry sight. Sometimes I'm sure you've gone in some rural parts of AARDO countries and you've seen scenarios like this. where it looks like a field has been abandoned, but it's sometimes due to circumstances beyond the farmer's control. So one of them is competition. The weeds will compete for the same resources that your crop needs. Remember, your crop needs optimum of these resources for you to have high yields. And this high yield here, there's many things depending on it. You want high yields because maybe this is you want to sell some of the crop, or maybe you are a subsistence farmer. This is where you feed your family from, or maybe you have even employed some people on this farm. You are relying on sales from some of this crop to pay salaries. So this isn't something that any farmer wants to see on their field. So competition. The second thing is parasitism. So some weeds are parasitic in that they actually derive their nutrients. They live on your plant and they are drawing nutrients from your host plant. And your crop plant absorbs nutrients from the soil and water and minerals, and it also undergoes photosynthesis and it produces food. All that is supposed to be used to create yield, all that is supposed to be translated into a cob of maize or grains of rice or a bunch of bananas. But if all those nutrients are sucked by a parasitic plant, obviously you end up with losses. You could even have complete crop failure. I have seen fields where a field infested with parasitic plants looks just like this image above. And of course, this innocent looking weed here is Striga asiatica. What you see here is Striga hemontheca. And these two parasitic weeds, they are typically problems in cereals. Like I said, you might find them harassing your sorghum, your wheat, your rice, especially upland rice. You will find them harassing your maize and so on. Striga gesneroides is much more of a problem on legumes, on cowpeas, pigeon peas. You will find that together with the Alectra vogeli. It's also typically a problem on legumes. Now, let's investigate this association you see at the bottom. Remember I said it looks very innocent, but it's not as innocent as you think. Let's look at it closely and see what is happening. So, if you move with me from the left to the right, first of all, you have a plant here. Okay, that green plant would be your crop plant. And if you notice, there's tendrils that are coiling around this green plant. So this parasitic plant, what it would do is, there would be some seed here in the soil. Of course, the seed is not shown. There will be a seed of the parasitic plant here in the soil. When it senses that you have planted your crop plant, your plant has germinated, there is exudates that plants secrete into the soil for various reasons. And the parasitic plant will sense from those exudates that the host plant is around. So it will germinate. And when it finishes germinating, the tendril will come and attach itself to your crop plant that you've planted where you're trying to get your yield from. And once it successfully attaches here, it even detaches itself from the soil. Because remember, this parasitic plant doesn't need to absorb anything from the soil by its own. Its duty is just to germinate and then attach there. Now, it looks like it's coiling around the plant, and it looks like it's an innocent, relationship. But if you look closely, if we zoom into that portion there, this would be your stem or branch of your plant, your crop plant that you have planted. The parasitic plant is coiling around, okay, like we see there. But when you actually do look at a cross section, you notice that the parasitic plant is actually sending structures into your host plant. And it's through those structures that it is now sucking the water from your host plant It is sucking the minerals and nutrients that are passing through the xylem and are in your host plant. And every time your plant, your crop plant is undergoing photosynthesis to produce the food that should then go to grain filling, and so that you have your rice, you have your banana bunches, and so on. The parasitic plant is actually sucking those nutrients and the food. So what ends up happening is your plant now, it goes through starvation. All that food produced is no longer available to be converted to harvestable products. And then, and that's how your yield loss occurs. And there's some cascutas and dodder species of parasitic plants that are here. Now, let's come down now to another example, which is the one for Striga asiaticus, Hemontheca, Electra, and several others. So, like I said, it looks like this is just some innocent friendliness between your host plant, which is your cereal, and the parasitic plant. But if you look at it closely, underneath, under the ground, beyond what you can see, this would be a root of your maize plant or your rice plant or whichever crop you have. And you notice that the parasitic plant is also sending structures into the plant root. And through that system, it's absorbing and sucking nutrients from that root, like we said before. So in the end, you have these plants that are, sometimes they are yellowing, sometimes they are wilting, and so on, because all the nutrients are being taken to the parasitic plant, which is this weed in this case. Okay, so we've spoken about competition, we've spoken about parasitism. The other issue is what we call allelopathy. Now, let's walk together through this image to try and get to what we are talking about when we're saying allelopathy, which is one way also in which losses occur. Now, allelopathy has got two sides. It might be a good relationship, it might be a bad relationship. In this case, it is the bad relationship that results in the losses from your crop. Let's look at this. Let's assume this is your weed, a weed. And weed species, different weed species will produce different allelochemicals or contain different allelochemicals. So this plant here, the weed, let's assume this is a weed in your field, you have not cleaned it out. It produces allelochemicals through the roots, okay? There will be root exudates coming out of those roots into the soil. That's one way in which allelochemicals are produced. Another way in which allelochemicals are produced is that when these leaves fall to the ground and they decompose, some of those allelochemicals leach out of these leaves into the soil. Now, once we have these allelochemicals, depending on the nature of the allelochemicals, and also depending on the plant, the species of plant that is in the vicinity, you can have these allelochemicals either inhibiting growth of your crop plant, and therefore you see it's stunted, or again, it might be chemicals that are enhancing the growth of your plant. So many weeds that we worry about, one of the reasons why you see the weeds creating problems, it's because they have produced some chemical which is inhibiting growth, and there is many different mechanisms through which these chemicals do that. Now, before we move away from this slide, at some point, several slides down, we'll be looking at some of the methods we use for controlling weeds in different scenarios. Since we are on this slide, let me skip forward and talk about weed control using allelopathy. It's easier to deal with it while the image is still here. Now, so the fact that you have allelochemicals that can be produced from your weed to infect your crop plant means that we can use that allelopathy to also inhibit the growth of weeds. Remember, these allelochemicals, as they are produced, they are not solely designed to inhibit the growth of your cultivated crop plant. No. It depends on the species that they are interacting with. It therefore means we as humans, we can also use that technique of allelochemicals against the weeds. The only thing we need to find out is which plant species have allelochemicals that we can use against the notorious species in your field. So you go to your crop field, obviously you've been growing your crop year in, year out. You know the species of weeds that are troubling you. Maybe it's a Cynodon dactylon, maybe it was one of the broad-leaved weeds that are troubling you. You know the species. So all you need to do is now to say, which are the allelochemicals? Which are the plants that have allelochemicals that work against these weeds? Okay? Remember, it has to be allelochemicals that will work against your weeds, but they don't work against your crop plant. So you can look for those plants, and there's two things you can do. Once you've discovered a plant, it could be a cultivated plant, it could be a wild plant that you incorporate into your field that produces these allelochemicals. Allelochemicals that don't affect your crop plant, but they affect the weeds. So what you do then is, you now go through there is a shorter, quicker way to do this, and there's a longer, more perfect route to do it. The first thing you could do is go through the process of a much more rigorous scientific process to try and understand what is this chemical and moderator, could you help me? Yes. Then you could go through the process of identifying this allelochemical. Obviously, if it's a company that wants to make a proper product that can be applied, they would go through that process now of product development, try to isolate, to extract the allelochemical. First of all, get to understand which is the allelochemical in this plant. It might be a native plant, it might be a cultivated plant. And then how do we extract it from this plant? How do we purify it? How do we bottle it? How do we package it? There's that whole process that goes into that. But there is also something that I've seen some farmers use. Once you've identified what plant it is, you can do two things. You can either plant that producer, the donor of the allelochemical, integrate it as an intercropping or mixed cropping in your field, so that as it is growing there, you have this allelopathic donor and you have your crop plant. So the allelopathic donor is continuously releasing these substances into the soil that are controlling the weeds, but your plant is going fine. Okay? That's one thing you can do. And there's many ways in which this is used. It could be through a crop canopy, it could be, and we'll get to that later. But that's one thing you could do. Have a live plant, which is continuously producing these allelochemicals into the soil. You could also harvest the leaves, okay? Harvest the leaves, crush the leaves, soak them in water, and then go into your field where you have these weeds and just sprinkle that water in there. I have seen this happen with, there's a plant called Lantana camara. I hope I forgot to include some images of it here. Lantana camara. I have seen some, we did some trials and where we just got some Lantana camara, crushed the leaves, soaked them in water, you know, for a day. And then the following day we went and sprayed it in the fields. and it did control several broad-leaved and some narrow-leaved grasses. Now, something like that is a low-tech thing that someone can do. Obviously, there's issues there to say, what is the correct kgs of leaves? Do leaves have more of this allochemical than the seeds and the flowers and the stems and the bark and so on? There is all those questions, you know, and there is work that is actively going in different places, researchers trying to fine-tune this. But this is a low-tech thing that someone can do, especially if for many small-scale farmers that are, you know, that may be resource constrained and yet make their living from agriculture. So that's something that can be used. So, but what it requires is that we do understand which ones are the allelopathic plants against which weeds, and there's lists of tables from previous works that have attempted to show which plant species have some of these allelochemicals. And while we are still talking about allelochemicals, yesterday we did agree that there's 4 scenarios under which one might do pest management. There's a scenario where you have like commercial farmers in a monocrop that rely purely on pesticides, synthetic pesticides. They see a fungicide, they see a fungal disease, they spray a fungicide. They see an insect, they spray, you know, an insecticide. They see a weed, they apply a herbicide. There is scenarios like that. But we also know issues that arose from that. We also know that there's integrated pest management, which you can use a combination of different things which you can go through stepwise. We also know that there's organic agriculture. I will run through these things again as we go down, just to remind ourselves and to see how uniquely weed management plays out in those scenarios. Then we have under agroecology, which I also attempted to introduce yesterday. Now, just to get ahead of myself a little bit, in agroecology, what I did say was that this is where you try to mix in different systems. You try to mimic nature as it is. And you want to do your fit into nature, do your agriculture by fitting into nature without causing too much disruptions, but maintaining systems together. For example, we know in the soil, a soil is a system. There's multiple living things there. Even though you have your plant roots there, there's several things that work together and they might be beneficial to the plant. We know that an aquatic system, if there's a pond nearby and there's living things, that is a system. You don't want to just put your paddy rice in there and kill off everything else that's in that pond, okay? A forest is a system. There's multiple species of trees and they interact. One of the reasons why, if you look at a natural forest, one of the reasons why you notice that you are not finding one species of plant obliterating all other species of plants. You're finding them existing, coexisting. One of the reasons is that the equilibrium is handled through things such as allelopathy. So if you are going to feed into an agroecological way of producing a crop, where you have livestock in there, and some of the dung from the livestock is going into your fish pond, and maybe, you know, you have your tilapia or some fish in the fish pond, or you have paddy rice, or you have something like that, where you are integrating these systems without really clearing forests like the commercial monoculture does. You have to be mindful of these interactions, and allelopathy is one of those that could be used for that. I noticed there is participants who are using pen and they are writing on the screen. Could you please avoid that? I don't know how to erase the pens. They might confuse other participants. They are looking like they are part of the illustrations when they are not. Okay, thank you. Now, So we've spoken about competition, parasitism. We've spoken about allelopathy. Here is another problem that could cause losses, from weeds. You can have your cotton plant, and when you are harvesting your cotton plant, you don't want this cotton plant. You want, at some point you're going to separate this lint for sale. If you have weeds such as the Biden's pilosa, obviously it sticks to your wool, and whoever you're going to try to sell this lint to, they'll ask for a low price because they have to go through a process of removing all these contaminants from the cotton. Okay. For those of you that have done manual harvesting of some crops, sometimes you will get into the field and there is this, you might find a weed there that irritates the skin. As you are walking through the field trying to do your harvesting, you will really be, your skin might have, undergo irritation. And when you're trying to hire people to help you harvest in a scenario like that, they might charge you more, okay? So that's an issue. Now, also, I have spoken about mechanical weeding, and we'll mention that hand weeding and mechanical weeding. For most farmers in many of the AARDO countries, many of the Africa and Asia, they make decisions about the size of land they are going to cultivate this particular year, mostly based on what they can afford to weed. Weed control is a major feature, and much more specifically, hand weeding and mechanical weeding. So when they notice that they will not have enough manpower to do the weeding, they end up cultivating only a small piece of land. Because if you cultivate a big piece of land and plant and you are unable to weed it, you are wasting time. So this is an indirect way in which weeds become a problem. Okay. Dr. Mfongwe, are you there?
Yes, I'm here.
I don't know
yes.
whether you can see these markers that are being put on the screen. Can you see them?
Yeah, yes, I can see them.
There's a participant who's putting them there. I know maybe it's there, it's happening accidentally. I do not know how to remove them.
Remove them. Yeah, that's the problem.
Yes.
I also don't know. I don't know. Let me check around maybe.
Dr. Just, I just request you to prepare to make me the host so I can just look it over there.
Okay, all right.
Okay. In the meantime, we can continue. So this table here is an example of weeding requirements, and this it may have changed now, it needs a little revision, but not by much. But you notice, and the two columns are important. You have the total column, which is the number of hours per hectare per crop that are needed to do, that are spent on a crop. For example, this is a maize crop, and in Ethiopia, on average, I noticed the recording has stopped. Maybe I should pause a little bit.
Yes.
I know it's probably because of switching hosts. I'll pause a little bit so that it's.
Okay, I think it has started recording. You can continue.
We can continue.
Yes.
I was wondering whether it's possible to make multiple people co-hosts. Okay, so let me continue. Like I was trying to illustrate here, you notice that to grow a crop, certain number of hours will be spent on land preparation, certain number of hours on weeding, harvesting, and let's say you spend 72 hours on that. You notice that for maize in this country, up to 54% of the hours spent on crop production They are spent on weeding, which tells you how much effort is needed to get in to do that weeding. And if you go down the column, you notice that the lowest is probably weed control is probably cassava, where it's only 25% of the time. But even this 25% is still a high amount of time spent. So because of that, you will have people shying away from getting big pieces of land for crop production. Okay. So let's now get to the second element of our talk, which is nematodes. So this is a big image, and I've blown it up just so you can appreciate the complexity of these nematodes, these worm-like creatures. Now, they are mostly in the soil. Of course, there is some big nematodes and there's microscopic ones, but the ones that we worry about in agriculture, it's usually the microscopic ones. They might be in the soil, or they might live as parasite on your host plants. Now, the problem with these nematodes is in the soil there where they are, their interactions with the plant roots are resulting in unhealthy situations. Decay of plant roots, swelling such as this. So what you see in this image, these should have been normal roots of a bean plant. This here is the stem of a bean plant, and going this way would be the above ground parts of the bean plant. This would be your soil surface, and everything this side should have been roots. But you can tell from these swellings, obviously, that something is wrong, okay? And so these roots will not be able to absorb water correctly, and the plant will not function well. In some cases, even up to 90% crop losses have been observed with some nematodes. You can see here, you have some maize plants, and then you have scanty patches where there are no maize plants, and you notice there are different heights. This tells you that there is some problem in this patch. There's a pest here. There's either a pathogen. In this case, this was a nematode. So if you see irregular stands, okay, thin irregular stands with stunted plants with varying heights, typically this is an indication that there might be a nematode. Thank you very much, Kamal, for removing those lines. Okay, so here is a similar scenario for the rice plant. You can see this is your rice field. You notice patches of this rice field are thin. Okay, you go to those places and you pull one of these plants, you notice there's swellings on the roots, and this is because there's nematodes. Now, coming back to now the overall idea of pest management. Like we saw in the discourse we had yesterday, it could be that some people would apply synthetic pesticides. There is many things that happened, which I'll repeat, that led to the introduction of integrated pest management, which could be weed management, it could be disease management. The principles are the same. Okay, the steps through you go, them are the same. There could be now you could be having issues with how to handle weeds in organic agriculture. Like we have said before, it presents unique challenges that require you to be mindful of how you do the control. Okay, just to recap again what we said before about the challenge with synthetic pesticides. Most of them are very toxic, and when you have your crop here and you're using those synthetic pesticides, okay, you could have them as fumes. If there's a person nearby who doesn't have protective cover on their nose, they could be inhaling these. They could be having these, could be making contact with the skin and getting into the person's skin dermally, and affect health. Some of these could be getting into the soil, okay, and they are interfering with the, a soil in the living system. You have many nice microbes there that help your plant. Like rhizobia, nitrogen fixing bacteria that you don't want to kill off. But with some pesticides, synthetic ones, sometimes that happens. Other times they end up into groundwater. You sink a borehole, you end up with drinking this contaminated water. And they can even either run off into a water body or through groundwater end up in a water body. And that affects several other things, marine life in there. We did go through the potential harms that could happen to a person, so I'll skip those. Okay, I just thought I should bring this out. One of the very commonly heavily used herbicide is glyphosate or Roundup. And there's been several studies that have shown, for example, it says here, up to 41% of people that were exposed to Roundup ended up having a Hodgkin's lymphoma. And there's papers that talk about all sorts of problems associated with Roundup. Okay, and recently there was a 200 million pounds or dollar lawsuit that was around Roundup. Okay, so these are some of the, yeah, I did say it could contaminate the environment, groundwater, it could contaminate food. If somebody sprayed their apples because there was a disease there, or they were trying to spray a herbicide to kill off the weed, maybe that herbicide ended up on the maize crop, the food gets contaminated. The non-target effects, you know, there's other living things in the environment. And when you're killing your weeds with the herbicide will not decide to only hit your weed. It will hit anything else that is nearby. Of course, you have development of resistance, and the costs associated with weed management are quite high. I have shared this image below, which shows that and this is an example of DDT, but there's several other pesticides and herbicides, by the way, that could bioaccumulate from one trophic level to another, causing problems. Even the person eating this fish eventually gets exposed to that herbicide. Now, there's an issue of herbicide resistance. I'm sure you may have heard about this. Where, because you are repeatedly using this herbicide, like glyphosate or Roundup, there's many reports of resistance developing against Roundup or glyphosate. And that could be because of your periodic or repeated use of this glyphosate. You are exerting a selective pressure on the weed population, so that the few that had natural resistance, but they were being kept at low levels by other biodiversity, they end up coming up and they increase in number. And all of a sudden, you use your Roundup and it doesn't work. There's many places where people are trying to use Roundup and it's not working because of that. Okay? There's also been some concerns that one of the things that technology that goes hand in hand sometimes with the use of Roundup is Roundup resistant crop varieties. So maybe you want to use Roundup in your maize field. So you insert a resistance gene, and this is a GMO, in your maize variety. Then you plant your maize. When that maize grows, the weeds are growing in there. You go with your Roundup and spray the whole field. Of course, your maize plant has a resistant gene, so it will survive, but everything else dies off. So there's been a concern that some of these Roundup tolerant genes that are put in these hybrids, GMO hybrids, might end up in, you know, local varieties, local addresses, and so on. Okay. Then another possible way could be, you know, maybe somebody accidentally moved a plant or an individual that has herbicide resistance from one place to another, and then it ends up multiplying. Before long, you have this resistance showing up. Okay, we did talk about pest resurgence, which is that these herbicides, when you use them, they might kill natural enemies. And as they kill natural enemies, the natural enemies should have been there to control your pest. For example, a wasp, many wasps are natural enemies of our pest plants. You kill off the plant, the wasp, you discover that that pest which was being controlled by the wasp, all of a sudden it shoots out. And the toxicity and effects on human health, I think I mentioned that. Now, we did go through this also. The idea of how did we get here and what is the nature of the situation we are in. One of the issues is monocultures, like you know, and we've discussed this over the past presentations we've interacted with. Sometimes, you know, a good example is Green Revolution. To quickly deal with hunger situations, not sometimes, it has benefited humans to get into these monocultures. You produce a huge hectareage of just one crop, maybe it's a staple crop, but of course, even in commercial crops that are exported, this is also happening. What happens is that this monoculture is deviating from your natural system. That's why systems like agroecology are trying to get back to the natural system, because the natural system used to have mechanisms of keeping pests and beneficial organisms and everybody else in equilibrium. Now, when you have monocultures, you have no rotations, you have no mixed croppings, and you don't have any diversity around. You don't have any other plants. You have the pests that used to be controlled by all those other living things. All of a sudden, they are free. There is nobody hunting them down. And so you start having problems of increased pests and diseases. And in response, because your monoculture, your 200 hectares of rice, of maize is being threatened, your response is to go and quickly look for some synthetic pesticides to blast in your field so that you keep your, people with food. And then the cycle goes on, and the pesticides, of course, they go and eliminate even more natural enemies. You have more resistance and so on. And then the cycle keeps going. Okay, so to break this cycle, we need to do some things. Okay, so that covers the synthetic pesticides and why we moved away from that. Now, let me quickly talk about integrated weed management. Again, I'll not spend a lot of time on this because we went through each of these objectives, what integrated pest management or weed management tries to achieve: effective, environmentally sensitive. Think about combining 2 or more methods. Understand your enemy as you design this. Think about when is it economical to do your pest management, and be mindful of the environment. Okay, and again, even in weed management, integrated weed management is better than just trying to hammer your weed or your nematode with just one method. We go through prevention, you make your observations, and then you intervene. What those 3 simply mean is this. Prevention, we've heard this several times. It's better to prevent something than to wait for it and have to firefight it, okay? We heard about when the whole world was struggling with COVID, it was helpful to have, hand sanitizing, cover your mouth, get through vaccination and so on. Those things, were being used as preventative so that you don't have to deal with the issue. The same thing happens with the weeds. There are certain cultural practices we can do, and I'll run through them shortly, that we can use to prevent or to reduce on weed problems. Now, the second principle, which is observational monitoring, like I said previously, if you are going to control a weed effectively, you shouldn't wait until it's overwhelming your field. You need to have a clever process of continuously monitoring so that you take action before the situation gets out of hand. That's typically what this is about. Also, the other issue with monitoring is it helps you to think about when is it going to be economical for me to take action, okay? And that's where we're talking about now action thresholds. And I did explain to say, for every organism, for every weed, if you have only 3 weed plants in your 10-hectare field of rice, that's not a reason for alarm. You could just probably pass through there and approach them and everything is okay. But as it starts to grow to a level where you know, if I don't take action, it will cause, it will be a pinch economically in my pocket, then that may be a time to take action. Okay, then when you get to now taking action, again, we ran through this, so I'm going to move through it very quickly. When it's time to taking action to control your weeds, because you've put in preparatory measures, you are likely to have less of a weed problem. In case those preparatory prevention measures didn't work, and as you monitored, you noticed that the pest was increasing anyway, then you intervene. The good thing with intervening when you have started by putting prevention methods and when you have already put in a monitoring system is that you intervene early enough. And it's very important, we all agree, that you have to tackle a problem early enough for you to be on top of it. So that's what this is about. Okay, and the major emphasis is don't start with pesticides, start with the friendly methods first, okay? So then the components, the actual steps of an integrated weed management requires that, of course, like I said, you need to establish, understand the densities that are acceptable, and then deploy preventative cultural measures, monitor, we've already talked about that. We'll get into how to use physical, biological, and chemical control options, if you must. Okay, now, the first component, which is establishing acceptable infestation levels. There is, among weeds, there is there's types of weed we call noxious weeds. Those weeds, like some of the parasitic weeds like striga, those weeds that are very difficult to control and they cause a lot of damage. Those are the ones we call noxious. For those, the thresholds for you to act, it would be very low. When you see even a few, it might be time to act. Now, there's several other ephemeral weeds that don't cause that much damage. You might see them and you can even sometimes ignore them until you notice that they're getting to. So for those kind of ephemeral weeds, your thresholds will be different. Okay, there's a whole list here of things that you can do to preventatively deal with weeds. And one of the things we said was clean planting materials. Sometimes the seeds of the weeds, they are so small that you'll be having your rice fig seed. And inside there is also striga seed and you can't see it. So when you have a proper clean seed, it helps you to deal with that. And there's several others, of course, time of planting, mulching, flooding, decoy crops, crop rotation. I've talked about the role of the diverse mix of crops and how that plays into your weed management. You can plant resistant varieties. There's some varieties that are known to have resistance against nematodes. Not so much weeds, but for nematodes, yes. You could do quarantine. If you notice there's a striga weed in a certain part of the country, you try to restrict movement of certain things from that part of the country. Or if you notice the neighboring country is struggling with this weed, you can put some quarantine measures to make sure that it doesn't spread into your country, and so on and so forth. This is an example of crop rotation. Up here where you have nice green healthy soybeans is a scenario where maize is grown one season, the following season groundnuts, and then the following season you grow soybeans. In a scenario like that, soybean cyst nematodes, since they like to feed on soybeans, but they don't survive on peanuts and maize, or groundnuts and maize, during the times when the peanuts and maize are there, you notice they reduce. But here is a situation where year in, year out, you're only growing soybeans. So you have a buildup of soybean cyst nematodes in this soil. Okay, so to help you with the monitoring, for the nematode parts, we have explained this. You walk through the field and you look for those symptoms. For the weeds, you'd have what we would call a quadrant that you randomly throw into the field, and then you count the number of weeds as a way of monitoring to figure out whether the density is increasing or reducing. Okay, the other thing you could do for nematodes, obviously, if you have patches, irregular patches of land that are bare because the crops have died, a quick way to assess that would be to pass a drone over it, taking images, like we discussed before. Obviously, the dry patches, because the dead patches, because of the nematode, you will be able to see from the images. And that's a better way of monitoring. Now, like we also discussed previously, there is a database and there's several others, digital tools that can help in this process of weed identification, but also identification damage done by nematodes. Kirby has graciously put up one such database, and there is even a mobile app that you can install to help you with that. For lack of time, I would have wanted to demonstrate that, but the time is not with us. These are just the examples of how such things operate. A plant clinic organized by Kirby. Again, there is modules you can use to train farmers and community leaders on how to identify weeds, okay, and several other pests and diseases. Now, when it's now time to, if you from your monitoring, you've noticed that all the preventive measures you've put in place, they're not working. The weeds are still increasing. Then now you intervene, and you're throwing in a physical control, mechanical control as your first method. Of course, if you notice there are only a few weeds, which is typical of what would happen if you're monitoring early enough, you can go in there and do hand, just pulling them out by hand. Now, the pulling by hand depends. There are some weeds like Cynodon dactylon that will have rhizomes and stolons underneath. Even if you yank the above ground part of the plant, you still have those propagative parts underneath. And those might be a problem because they will just regenerate back up. You slow down the weed, but they will regenerate up. Moderator, may I suggest that I continue and just finish the slides before we switch to questions and answers? I know I'll be done by 12. Is that okay?
Yes, it's fine. You can go ahead.
Okay, so that's from 12 until the time for the valedictory section. We can just be dealing with questions and answers and comments and discussions, since we have experts that have lots of experiences. Okay, so like I was saying, when you notice there are very few, hand pulling might be a good way to go. Of course, on condition that this is not weeds that have underground propagative structures. Now, one of the other crucial things with, of course, you can get into mechanical control. And here you're talking about some implements that may be powered by a battery or by, some kind of mechanical. It could be a tractor, it could be oxen, it could be water buffalo, and so on. Now, it is very important that you do your control, your mechanical control, before the weed sets seed. When the weeds, if you wait until the weeds have flowered and they have produced seed, even if you go there to do mechanical control, the seeds are going to fall into the soil. And what will happen is you're going to have this soil seed bank. In the soil, you will have a lot of seeds. And so they will just remain dormant. What will happen is every year when you come to cultivate and plant your crop, some of the weeds will germinate from that seed bank. So for you to deplete the seed bank over time, you must make sure that you prevent addition of seeds, weed seeds into the seed bank. And you do that by making sure you deal with the weeds before they flower. Okay. Sometimes weed seeds and weed propagules are moved from one place to another through machinery. So this actually should have been a preventative. We should have moved it to the preventative control measures to make sure you don't move weeds from one field to the other. Okay. Now, one other physical method that you could use is what we call solarization. Like I said, when we're talking about pathogens, and of course, we are still talking about one pathogen, nematode here. The category of things we are dealing with here, the nematodes and the weed seeds. Sometimes by doing solarization, that is, you put plastics, you water some, moisten some soil and cover them with these plastics, maybe for 2 weeks, 3 weeks, a month, depending on the persistence of the microbe you're trying to kill. You will find that the temperature build up underneath there will not be good for nematodes. Okay, it might kill off some nematodes. Of course, there are certain cysts, nematodes that are in cysts. You might just reduce their numbers. You may not completely eliminate them, but that at least helps. Okay, and sometimes some of the weed seeds would be killed by that high temperature. Okay, it's also possible that the weed seeds that have received, because you put some water in there, some of the weed seeds will absorb water and start to germinate. But after they germinate, it's like suicide germination. After they germinate and there's nowhere to go, the temperature is high and they're covered, that's how they end up dying off. And that way you are reducing your nematodes and your weeds. Okay, so the next component, Dr. Zahara, could you kindly check the way you use your cursor? I think you did add a line to the screen. Thank you. So like I said, your first is preventative in IDM or integrated weed management. And then of course you do your monitoring, and when you start to intervene, you go first to the physical methods. Then the next line of defense would be biological. And biological control requires a little bit more preparation. If you've noticed from your previous seasons that we are having a recurrence of this pest problem, it might be time to start adding on biological control onto whatever method you're using. Because, for example, if you have a weed, it keeps persisting, year in, year out, it's persisting. It means even though you are destroying it at the end of each season, there is still a lot of seeds in the seed bank, in the soil, that is not attended to, that needs to be dealt with. And putting up biological control ahead of time might be a way of making sure that by the time the weeds germinate each season, the microbe or the insect that feeds on that weed is already present, okay? And it controls it. So for weeds, we can have microbes, we can use allelopathy. I spent a lot of time explaining allelopathy. We can have insects that can feed on your weeds. Of course, make sure these insects, you have to make sure these insects are not feeding on your crop, but they're only feeding on your weeds. Specificity is important. And there's cases where fish and ducks have been used in aquatic systems to control weeds. For nematodes, under this biological control approach, the main things that are available, that are popularly available, is microbes and plant extracts. And these are areas that have lots of potential that we should look into. Of course, the preparation, the identification. So you could either import these natural enemies from overseas, or you could do your bioprospecting to try and look for those that are, adapted to your country for introduction. But of course, in the beginning, it might require some capital outlay. But once these systems establish, you may find that from season to season, you may not need to, you will have some of these microbes beginning to establish, or your insects. Okay, now, as examples, using microbes. This Piscilomyces lilesinas is actually, there's actually a commercial product that's available in some other countries like India. So for you folks that are trying to look into use of biological control of some weeds and nematodes and so on, please look around your markets. You might find, you might be surprised. Some, I know, like in India, they do have some. And some of the products in India, I think I've even started reaching other countries. Okay, so there is options for root knot nematodes, and there's several other examples. And I think this would be a very good way, especially if you can get this Piscilomyces lilesinas, or the Verticillium lecani, or the Bacterium penetrans to establish in your countries, in your fields. That would become a very it becomes a good method for long term, and becomes a method that you can easily integrate with other systems. Another good example is Fusarium oxysporum. And this, again, it has received increasing use the recent past. And in Africa, we had Kenya. I was listening to the news some 2 months ago where Kenya was actually registering this for use. And there are several other countries, including Zambia, that are currently trying to identify our naturally existing Fusarium oxysporum strains for the management of strike and several other things. So there are several other options available here. And depending on your country, you could actually, when you have weeds, you could actually look at your weeds. If you have a setting where there's no crop, you could actually look at your weeds to see what diseases the weeds are suffering from. And you could try to spin something from that for use. Okay, in terms of insects, there's, again, several examples of what could be used. Lantana camara has been effectively controlled previously. This is not to say this is the only insect that Dr. Zara by something. Could you kindly check the way you're using your cursor? You are adding lines to the presentation, and they might interfere with for illustration purposes. Thank you. Okay, so that's with insects, and you can actually, if you have certain weeds that are problematic, if you do a survey, you could either try to import, insects from people that have already dealt with this kind of thing before, or you can have some entomologists looking around doing surveys to see what are the natural enemies of this weed around, and that could be the basis for biological control using insects. I was mentioning the management of aquatic weeds. Paulina acuminata, this was an insect that was introduced into Zambia to deal with the Salvinia molesta. And it did a good job. It ended up being tried in other countries also. So if you are thinking about a natural ecosystem, like if you're thinking of controlling weeds under agroecology, for example, and we'll come to that, instead of blasting chemicals, all that, you can find a way of integrating these insects into a system like that. We'll come to that again. In terms of others, here you could have cattle, sheep, goats. Of course, they have to be cleverly integrated in such a manner that they don't feed on your crop. Fish has been used, and the grass carp appears to be one of those versatile ones, successfully used in Egypt. And what we know from literature is that it feeds on a broad range of not aquatic weeds, not all aquatic weeds, but a very good range of aquatic weeds. So this is something that could be integrated if you are doing organic agriculture or you're doing agroecological crop production, or as it may be. Okay, so still under biological control, you could have live mulch. And the live mulch simply means you have a plant, a live plant that is in your field already, and then you come and add your crop plant in this live mulch. So what happens is this live mulch is typically a plant that has allelopathic effects against your weed. So as it is shedding some leaves, those leaves might go and, or maybe the roots are producing these allelopathic effects, but it also could be that it's just the actual mulch, dry leaves falling off and covering the ground. If you have dry leaves covering the ground, you end up with the suppression of weeds, because the weeds that are underneath, the weed seeds, when they try to germinate, they don't access sunlight because of that cover. And so you have weeds, being controlled in an environmentally friendly manner. I've talked about allelopathy, and I should mention also antagonistic plants. Some people may have heard that if you have marigold plants in your field, and again, there are different species of marigold, you have to look into the exact one. They produce exudates in the soil that deter nematodes, or that interfere with the growth of nematodes. So this might be a very good way of combining this into an agroecological way of handling nematodes. Integrate, as you have these natural systems in plants in there, look for plants that you can add in there that have this effect on your nematodes. Okay. Then chemical control. It's usually difficult to run away from chemical control, and we have discussed the reasons why. And what we unanimously seem to agree on is that short term, yes, but long term, we should think more of planning, deploying better IDM systems, or trying out agroecology and organic farming. Should you use chemicals, please don't rush to the very toxic ones. If you are going to integrate chemicals into IDM, try to start with the slightly hazardous ones. Okay, so now I've been mentioning organic agriculture and agroecology in passing. Let me just refresh our memory on what the expectations are from organic agriculture and agroecological crop production. That way we are on the same page, and we all agree when we say you cannot use the pesticides in organic agriculture and agroecology. So organic agriculture looks at a broader, it's similar to agroecology, but a little different. The major emphasis there is, of course, nature, although there isn't an active attempt to connect the different systems, okay? But you want to try and mimic as much as possible the natural way in which organisms exist. And in that manner, moderator, could you help me? In that manner, you don't want to introduce a lot of these inputs that might have adverse effects. Pesticides, we've talked about their issues. Synthetic fertilizers, there are certain issues that have been, noticed in synthetic fertilizer use that are thought to not be consistent with organic agriculture. Okay, so just to run through that quickly, the principle of health. You are looking at health collectively, an organic agriculture system. The health of the soil. It is understood that if you feed your soil well, you feed your plants and your animals and the humans, you have overall good health. And because of that, you end up not requiring to worry too much about, numerous pest control management. The principle of care, the fact that the realization that the environment is fragile, it's not endlessly resistant, it could be damaged. There is, the realization that, the environment is fragile. And because of that, you have to be mindful of the practices you introduce. They must not harm the environment. That's an important principle. There's a principle of fairness, equity, respect, justice, stewardship towards resources. All living entities in a given environment, they are beneficiaries of that environment. So everybody's needs must be met. There is so that's one of the principles. And of course, the realization that in every environment, in these agroecologies, there's system. The soil, there's the soil system. In the aquatic system, there's forest and so on. So we must not disrupt those. Those are the tenets. And if we contrast that with the conventional way we've known to deal with for a long period of time, the synthetic pesticides, we notice that the chemicals are not in agreement with the principle of health. Okay? Still, the principle of care, where you're not supposed to damage the environment, we know that chemicals can damage the environment, and therefore they're not consistent with the organic agriculture. There's some exceptions where something is really, really desperate, where exemptions have been made in some cases, not globally. Okay, there are some countries where they might agree, as you would understand, there is organic agriculture and there is a few modifications to it. Okay, and this is one of them. There is some countries, and one of the participants, I think it was Dr. Farouk, did mention some exemptions. So there is exemptions for a few chemicals, but in general, the realization is that chemicals, synthetic pesticides affect nature, and so they should not be included. And you'll notice even among those chemicals, it's probably those that are least toxic. Okay, hybrids and the varieties, certain varieties might be restricted access. So you'll notice that in organic agriculture, they are very mindful of, especially GMO varieties are not allowed. Okay, and then the conventional system, monoculture, for example, it comes to an environment, you clear everything, obliterate everything that was there, and you only put one crop. So that ignores the ecology. Okay, so let me run real quick to agroecology. And like I showed you last time, you have systems. Here you have water, you have your rice, you have mangoes in one system. You have a whole array of different vegetables here in one system, with plants and some planted trees and some naturally existing trees. So you see that there is very minimum disruption of nature as it were. Okay, this is a similar situation. You have different species planted together, and then you have some planted ones, you have some naturally existing ones. Okay, and here is another one where you have you have your pond, you have fish in here, you have your rice, you have some of your effluent from there getting into there, and you have your nature around. Okay, and so the issue is systems based. Okay, that's very important. Systems based in response to it, in relation to what we see in nature. Take many systems and integrate them in a way similar to what we see in nature, which means that you have to think about this ahead of time. You need to design it. You need to design the systems because your house is also there on the same piece of land. You're going to have goats there, maybe. You're going to keep your maybe paddy rice. You're going to have some. So it means that you need to, if you're going to mimic nature, if you go in nature, you'll find in a forest, in the middle of the forest, there's a stream or there's a lake and there's forest and all these. So that's the whole idea, okay? And the thinking is that some of the principles you notice are similar to what is in organic agriculture, but the difference is that systems things, integrating systems, and designing that system ahead of time. So biodiversity, very important. Efficient use of resources, instead of the cow dung being thrown away or the chicken dung being thrown away, it can be used in a vegetable garden, in a fish pond. Resilience, if you have many different crops there in that system, if one crop fails, you will find food from another source, okay? Co-creation, the realization that anything you develop must be co-owned, must be co-created, and the knowledge must be shared in that community, okay? And there's several others here that I'll not go into. I've highlighted these in green because these are the ones that are closely tied to weed management in agroecology, okay? So like I said, again, the difference is mostly because in agroecology, you have these planned interactions or integrations of systems. That's mostly how it differs from organic agriculture. So now, if we examine all those things we have been discussing under general idea, we will notice that these are things that must not be, that will apply for agroecology and organic agriculture. A lot of those preventative control methods we discussed, okay? Just because you are doing organic agriculture or you're doing agroecology doesn't mean that you can use any seed you want. No, make sure this seed, if you got it from your neighbor and you're coming to introduce it into your agroecology or in your organic agriculture, at least make sure this neighbor of yours didn't have a parasitic weed, didn't have a heavy weed infestation. So you know their seed is clean. So you can still merge that into IDM, into agroecology if you want, or into organic agriculture. Of course, the use of genetically modified organisms is a no-no for agriculture, is a no-no for organic agriculture. And remember the example we talked about here is you can have a genetically modified maize variety, for example, which tolerates glyphosate. You plant your maize, weeds grow, you come and spray the herbicide glyphosate to kill the weeds, leaving your maize alone, okay? Then there's a whole array of mechanical and physical methods we discussed, and these are very amenable to agroecology and organic agriculture. Of course, you have to select here because you're trying to maintain this diverse reach of mix of plants. It doesn't mean you go slashing every other plant around. Just selectively remove those that are encroaching into the spaces where you probably want your crop plant to be. Biological control, in that rich mix of in an agroecology setup, for example, you could introduce plant species there that have natural allelopathy and so on. I think we went through a lot of those, okay? So in conclusion, there is all these options and there's a lot of the old methods that have been used before, and there are several new things that we have observed happening that we could enhance on. Remember, integrated disease management is still a very, very important concept. So we could borrow some of the improvements we are noticing that the groups in organic agriculture are talking about, the groups in agroecology are talking about, and bolster up the integrated disease management, the integrated pest management, and the integrated weed management. And if we do that, we should be able to improve our yields. We should be able to deal, to correct, to avoid mycotoxins. We should be able to limit crop damage. I thank you very much for your attention. I see it's almost 12 hours now. It's 2 minutes to 12, I think. So over to you, moderator.
Okay.
Thank you very much.
Okay, thank you very much, Dr. Paul, for the presentation. So we can go into question time now. You are free to ask questions. Let me see who is there. There's one person.
By the way, Dr. Mfungwe.
Yes.
Not just questions, even comments are welcome. There is unique experiences that we might learn from some participants. Thank you.
All right. I hope you've gotten that. So questions, comments, clarifications, you're free to bring those up. We are here to learn from each other, from your experiences as well. So we've got two people raise their hands. So Dr. Sabine from Pakistan and Yusria Ibrahim, you can go ahead with your questions, starting with Dr. Sabine.
Yes, thanks, Dr. Paul, for your excellent lecture. I have the question, can we use leaf extracts of Lantana camara against narrow leaf weeds of wheat? And is this practice is going on in Zambia? Second, I want to know about the control of Leptocloa chinensis. Even using the chemicals, this weed is not under control in Pakistan in dry system. And my third question is, what is the standard method of taking samples for nematode? Thank you.
Yusria Ibrahim, you can go ahead with your question.
Hello?
Yes, we
Hello.
can hear you. Yes.
Can you hear me?
Yes, very clear. You can go ahead with your question.
Okay,
please.
my question is related to the use of microbes. When using the microbes or byproduct, there will be a time when those pests develop resistance to this type of IWM. This is my question. Thank you.
Okay, thank you. We can get one more question from Fanuel from Ghana. You can go ahead with your question.
All right, thank you. I'm so grateful to be part of this international session. Yeah, to Doc, the presented lecture. Yeah, regarding the use of Lantana camara, would you be in the position to let us know in terms of quantification, how much quantity that is required to be applied to the desired crop or the weed for control? Yeah, that's my question. Thanks.
All right. So, Dr. Po, I think you can go in with those three questions.
Thank you very much, Chair, Moderator. I did miss, I think, the second question from Sabine. I think I heard the first one, which was talking about Lantana camara. I think her question was in connection with narrow-leaved weeds. I think she was saying, I don't know what she was saying in wheat. And then also, she was talking about, I think, standard sampling for nematodes. I think I heard that one. But there was another one in the middle that I missed. Dr. Sabine, could you repeat the second one? The Lantana camara narrow leaves I heard.
Yes,
the nematodes I
yes. Yes, Dr. Paul. I was talking about the spread of Leptocloa chinensis. weed in our rice system. Even the use of chemicals has not, we don't get rid of this weed. And it is so much widespread that in our rice field, its growth is overloaded as compared to the rice.
Okay, if I hear you correctly, the question is what to do about it, the Leptocoria sinensis?
Yes, Leptocoria sinensis, wheat or serious weed in rice in Pakistan, that is not getting controlled by chemicals even.
Okay. So let me start with and thank you for your questions, Dr. Sabine, Ibrahim, and Fanuel. Let me start with the question relating to Lantana Camara. So with the Lantana, there is a lot of experimental work ongoing. So the work I was referring to, where we were noticing some control, and I can share I can share this publication with those that are interested in looking at it further. We did notice, certain weeds that were being controlled. Now, we tried this, I think, in sunflower, and I think we tried it in maize as well. And we were noticing, of course, that both narrow and broad-leaved weeds were being affected. So it would be important before we go full out and attempt to use it in wheat. We know wheat is also a grass. It would be important, first of all, to establish whether wheat is sensitive to the allelochemicals in the Lantana Camara before we use it. But the fact that it has affected both broad-leaved and narrow-leaved weeds, it's a good area for for further investigations. I think that's what I'll say with the Lantana. Concerning the quantities, if I'm to jump to Fanwell, the quantities to use, because this is a space where you have research ongoing, but also you have farmers trying out certain quantities. Obviously, when you increase your quantities, you will have more of that allelochemical in your field. So the more quantities, the better. However, we don't know whether excess amount of it might be toxic to the plant. And therefore, in terms of quantities, we can share with you, Fanwell, through that publication, the quantities, and I can even share with you with other people that tried it, the quantities that they saw that were effective. Obviously, the issue of the quantities is important because if you put too little, you might say the technologies that doesn't work. Okay, so you want to make sure you're getting good quantities. And also, when you soak, if you're going to soak your lantana, you crush it and you soak it. Of course, there's an issue of duration. If you soak it too long, you might reach a point where that allelochemical, even though it's leaching in water, it starts to degrade. So that issue is not yet known, but we can share the durations that we tried. But also, with the quantities, you don't want a technology whereby the quantities required for running the technology are so high that you cannot find enough Lantana Camara to use. So Lantana Camara, as you know, is an invasive plant species. So in many places where it's an invasive, it's easy to find huge quantities of herbage for use in this. But in the event that in your country there's little of it, obviously then it becomes helpful to you only if small quantities are required to control that. But as to the quantities, Fanwell, I will share the quantities we tried. Obviously, there is need to run a trial where you stagger numerous quantities to see which one would be the best to deal with. So that would be the issue to do with Lantana camara, narrow-leaved weeds in wheat, as well as quantities by Fanwell. Now, to get back to Dr. Sabine a little bit, that Leptochloa sinensis, which is you're trying all sorts of chemicals, and it's not going away. It's probably time to, obviously I know the physical methods will work, but again, if you have 5 hectares of rice, it's you know, between the rows of your crop, if you have planted your rice in some rows, okay, you could use some of those you know, mechanical tactics that I see. There's a slide where I put some of those mechanical, but I understand if you have a huge piece of land, mechanical control, you know, becomes quite an issue. So for now, I think the best way to go is continue with those mechanical control methods. Obviously, it's difficult to prevent it. I don't think we don't know whether it comes with a weed, with a seed, with a rice seed. I'm not sure what the size of the seed is, but if you are having your clean seed and you know it's not contaminated. No, actually, I think it mimics, tries to mimic rice a little bit. So if you can have your clean seed and you can, for now, run with those mechanical control. On a longer term, I would suggest looking into biological control. Of course, if you look into that, you have to look for methods that don't interfere with your rice, something that will feed on your wheat, but not on your rice. I am not very familiar with this weed, but I can double check it for you. What I know is it must have a natural enemy. If you can find a natural enemy that does not affect your rice, that would be a good way to go. I can look into this and give you some feedback later. Now, in terms of sampling of nematodes, of course, typically we go with the soil, but if you are going to do forecasting to try and figure out how much of a nematode problem you are likely to have this coming season, if that was the question you answered, yes, you'd have to go into the field before you plant. You will find it in the soil, and you want to go get random soil samples as rich and detailed the sampling as your funds can allow. But the monitoring in terms of integrated disease management, when you are trying to ensure you react before all your crop is lost, that is a lot more like what we do with many other plants. You walk randomly in a zigzag in the field, observing your plants. I think it's the best way to do. Because remember, even though your nematodes are in the soil, if, for example, there's some soil treatment you did before you planted, and that soil treatment didn't work very well, okay, you will notice that during the monitoring. As you move around in your rice field, you will notice that this nematode problem is still here. And you notice that problem by looking at the plants, okay? By looking at the plants. If it's in a rice field, for example, you will see those, you know, typically, you would, not just rice fields, most of these cereals. Like I said, you see these irregular patches of either stunted plants or plants that are not there, they are dead. That tells you that you might be having a nematode problem, okay? And in such patches of land, if you pull out a plant, okay, in such patches of land, if you pull out a plant, you will notice, you know, root damage. If it's, and you typically your root knot nematodes, you will see the knots on the root. But You don't go around pulling plants anyhow, because it becomes too destructive. But I think there is a way to combine both. If you begin to notice some damage happening somewhere by, again, going by the characteristics of the plants, you could combine that now with soil sampling in those specific spots. But as a preventative tactic, if you know that a certain field is normally problematic where nematodes are concerned, you could go sample the soil before planting, and then you can try to do some things, okay, ahead of that, either some physical solarization or some other kind of treatment. I hope that answers your question. Ibrahim, you're talking about using microbes to control weeds. So is resistance going to develop? No. So resistance usually doesn't develop from biological control. If a microbe is killing a weed, okay, unless you have genetically changed this weed so that it has started producing toxic substances that are killing the microbe, that is unlikely to happen. If you have a microbe that is killing a certain weed, it will always kill that weed. If you have an insect that is feeding on a certain weed, there is not a time when the insect will stop feeding on that weed. So that's the beauty of biological control using microbes. You don't have, because unlike where we do plant breeding, where we are selectively looking for alleles that we are combining into a plant so that this plant now has resistance to this microbe. That is not what happens in nature. In nature, microbes and plants, they interact in an equilibrium. The microbe doesn't act to wipe out the plant. So the selective pressure is not as strong as what we normally see when we're using synthetic pesticides. Thank you for those questions. Over to you, moderator.
Okay, thank you. Thank you for answering those questions. We go to the next 3 questions. The order will be Charles Kanyika, then Jesiah Kamati from Namibia, and Justin Muhango from Zambia. So those 3 can ask in that order, starting with Charles Kanyika, then followed by Jesiah Kamati and Justin Muhango from Zambia. Mr. Charles Kanyika, you can go ahead with your question.
Thank you so much for giving me this opportunity. I wanted just to find out concerning the IDM, integrated disease management, and especially on the weed. I've got the field now, I'm suffering from elephant grass. So now I wanted a recommendation, maybe you talked about it, noxious weeds. How do we control noxious weed? And what is the best method we can use? Could it be biological, chemical, or mechanical? That's what I wanted to find out.
Okay, thank you. Mr. Jesiah Kamati from Namibia, go ahead with your question.
No, I just want to get the concept solarization. No, is this more good for dry or maybe cold countries, or is more good at hot weather or cold weather? Because I want to understand the concept of solarization in more details. Maybe Doctor, can say more about that?
Okay, thank you. Mr. Justin Muhango, you can go ahead with your question.
Good afternoon, Doc, and thank you very much for the good presentation. I would like to find out on aflatoxins. My question first is, before I ask a question, I want to tell you the background of my questions. I'm an agronomist in Zambia, and we deal a lot with the small-scale farmers. who most times are maize growers, and they do not have access to fungicides. This means the inaccessibility to fungicides. It means that most of these maize, if not taken care of during harvest and storage, and sometimes even during production, they get the aflatoxins, which are actually very poisonous to human beings. I understand that by law in Zambia, only the maize that is exported out of Zambia are tested against aflatoxins. Now, what methods should be implemented or applied by these small-scale farmers in rural areas who are actually the majority in Zambia to control aflatoxins in the absence of fungicides? Thank you very much.
Okay, Dr. Paul, you can go ahead with the 3 questions to answer the 3 questions.
Thank you, moderator, for that. And thank you, Charles, Kanyika, Kamati, and Justin for your wonderful questions. To start with, Mr. Charles, the elephant grass and also noxious plants in general, how to deal with them. There are no differences in the way you deal with the noxious versus non-noxious plants. The major difference is this, that with the noxious plants, in the early stages of the integrated weed management, like we were discussing, you want to make sure that if your preventative cultural control methods have not worked, and during your monitoring, you notice that the noxious plant is still there, okay? What you want to do at that step is to make sure that you take action earlier. If you notice that, unlike ephemeral weeds, where you will see a few plants, one plant, and you know that it may or may not affect your yield. With that one early on, with the noxious weed, you act much earlier when you're monitoring. Even your thresholds are different. And in many noxious weeds, unless it has become so rampant all over the country, one of the things that tend to be applied to noxious weeds is quarantine. But I know sometimes the quarantine initially might be deployed, and then you still have the weed spreading throughout the country. So my advice would be that still go through the different steps of integrated disease management when you're dealing with that. If you have this field, for example, where elephant grass always shows up. Now, unfortunately, I don't know yet. I don't think we have in many markets a biocontrol agent microbe against elephant grass. I haven't heard of any. It might be there somewhere. I haven't heard of it. But my advice is just go through the regular integrated weed management steps, but start earlier. And so like that elephant grass, it has got lots of stolons and rhizome, stolons. So it's not an easy weed to control, especially when it has established in a certain piece of land. If it has established, you might kill the above ground parts, but the stolons are going to always be underground. Even if you go and plow and tend the soil, you reduce it, but some of the stolons, they'll still be there. And those stolons, even if you plow them and they're up and they might look like they are drying off, and a few would still be covered by soil. my advice is please just go through your regular IDM or your regular integrated weed management. Now, to explain a little bit now, Mr. Kamati, with the solarization. The idea with solarization is to increase temperature in the soil. So it's not something that is only used in the cold countries or in the hot countries or in this region or that region. It's a technique that can be used everywhere. The idea is you're trying to increase the temperature in the soil. Of course, it works best in the tropics because in a hot season, for example, in the tropics, the soil gets very hot. And so just naturally in the tropics, the soil tends to get very hot. So if you put that plastic there, it gets even hotter. So in terms of results, you will see a lot more effective results in the tropical environments where it heats up. But otherwise, it's a concept that you can use anywhere. The reason why you are adding some water to the soil before you cover them with plastics is twofold. One, heat transmission is much better when it's a moist substance. But two, that moisture, when you've added to the soil, what fungi tend to do is that the spores in the soil, one of the things that tells them that it's time to germinate now, the plant might be nearby for you to affect. Or one of the things that activates nematodes, or indeed weed seeds themselves, it's moisture. So by watering there, you are signaling to those things. Remember, the seed which is dormant is difficult to kill. You can kill it, it's difficult to kill. A spore which is dormant is also, you can kill some, but some are very difficult to kill. The same thing with some nematodes, like cyst nematodes that have a cyst around them, that put their eggs in a cyst. They are very difficult to kill. But when you put some water there, it's like you are telling them to lower their guards. They start to germinate thinking, if I'm to put thinking in quotes, thinking that the conditions are okay, thinking that it's now time for us to come out of hibernation. In the meantime, you're actually not planting anything. You just put water there. So that brings some activity to the fungi begin to germinate, and then they become more vulnerable to heat stress. The nematodes begin to activate themselves, and they become more vulnerable. The weed seeds also, the moment there's water, they'll absorb water and they'll start to germinate. And the moment they have the radical and the coleoptera shooting out, then they are even more sensitive now to temperature, to increasing temperature. So they are easier to kill. So that is, in summary, the issue with the solarization. You are trying to kill things that are in the soil and by raising temperature. Okay, like I said, when you have 10 hectares of land, it's difficult to use this technique. But if you have a high value crop that you're growing in a small piece of land, it's very easy to do this. Some people that are not limited, they even pass an electric pulse through the soil, sort of electrocute the nematodes. But that obviously is not something that's possible for a lot of developing world countries. Now, Mr. Mohango, with the issue of aflatoxin. Yes, I do agree with you that most small-scale farmers, the issue of fungicides is difficult to access. And therefore, any technology that heavily relies on fungicides is difficult for small-scale farmers. But just to mention a few things, the first thing is this: even if you have a fungicide and you have a maize crop that looks clean, no disease symptoms on it, there could still be a fungus growing there on the maize. You will not see it with your eyes. The maize cob is clean and nice and healthy. You can still have these mycotoxin producing fungi, and especially this Aspergillus for aflatoxin. Even when you don't see any damage on the plant, you don't see any damage on the cob, you could have the fungus there growing. So fungicides is not really a good option for management of aflatoxins. Also, you did mention testing. Yes, testing is an important thing, but that is at the terminal end of the maize production system. But also, the testing, you're talking about exporting and testing. Usually, it's not a law for us to test to export. It is the countries we are exporting to, the sensitive ones. There are some markets that are not very sensitive to aflatoxin regulations. There is others that strictly enforce and they regulate. If you are going to attempt to export groundnuts to South Africa, South Africa is very sensitive. They are going to test it. The Zambian government will not force you to test the groundnuts, but where you're trying to take it to South Africa, they will insist that we want it tested and it has to be free of aflatoxin. The same thing with the European Union. But, you know, when you're exporting to a place like DRC where there isn't very strict enforcement, you might find that they are not tested. Even locally within the country. Now, and in many countries, not just Zambia, many countries are beginning to increase enforcement for aflatoxin testing, because we realize that if something is so bad that you shouldn't export it to Europe or export it to South Africa, it means that you shouldn't even eat it internally within your country. So there's a increased sensitivity now. Now, in terms of methods that low-tech methods that farmers can use, there are several. One of the things that happens is, of course, your aflatoxin contamination, it starts in the field. When the crop is growing in the field, sometimes at the time you harvest your crop, it might already be having aflatoxin. And then it gets worse when you go and store your crop in a very poor condition. And then the mycotoxin and aflatoxin really builds up. So in terms of farmers, one of the things that tend to result in mycotoxin buildup is a stressed crop in terms of moisture. Crops that are grown in environments that have low moisture and high temperatures, those crops tend to succumb a lot to mycotoxins and aflatoxin. And partly because the fungus that causes aflatoxin in this case, it's a xerophyte. It loves such hot, dry environments. Meanwhile, your plant is struggling. It's barely able to defend itself. So in the least, if you're able to provide water for it, that helps, okay? 2, sometimes when you have insect damage on your crop, for example, on your maize crop, you create entry points for the fungus that causes aflatoxins. If you're able to deal with insect damage, you can help. I'm not saying this will solve everything. It contributes a small portion towards the reduction in these aflatoxins. 3, there is a biological control product called Aflasef. I think this is a real game changer. I know it was registered in Zambia and in many places in Africa and even in Asia. There are some places where they've developed this Aflasef product using their natural, it's a biological control product, using their naturally existing fungi, atoxigenic Aspergillus flavus. They have these. So you spray that in the field at about flowering time, okay? So it establishes in the field so that it competitively excludes aflatoxin-producing fungi. I know that in some countries like Zambia, it's been registered, and for Zambia in particular, right now it's undergoing commercialization agreements are taking place to say, how do we roll out this technology to farmers in such a way that it's available for the farmer, in such a way that it's not just for the commercial farmer. I think that would be a good one. Also, moisture content is very crucial. When you harvest your crop, you have to dry it carefully. Dry it carefully. Otherwise, if you take a crop that is not very dry into storage, that environment becomes humid from the moisture on the grain. The fungus begins to grow and the aflatoxin or mycotoxin builds up. So dry it properly. But also in storage, even if you're taking grain that has been nicely dried and you put it in storage, remember this grain already has some fungus on it. But so if you put it in storage and it's poor storage, it's humid, it's leaking, there's water all over the place, there is rats and things good, then that is the recipe for increase in these aflatoxins. So I think in a nutshell, Mr. Jester, we can chat more about this. It's one of the areas where I work closely in. Those would be some of the methods to use for small scale farmers. Of course, the testing is at the terminal end. If you test and you find there's aflatoxin, you're not helping the farmer because you are going to throw that crop away. You are helping the consumer, and that's very, very important, but it will not help the farmer. Thank you, moderator. I notice I've spilled over 12:30 by some 4 minutes or so. Maybe the valedictoria is about to start.
Dr. Kamau, can you advise if you are ready, we should maybe transition into the valedictory session? Hello? Okay, maybe while we're waiting, we can just get the last 3 questions from Mr. Muhammad, Mr. Emmanuel David, and Dr. Farouk in that order. So Mr. Muhammad, you can start your question. Mr. Muhammad, you can unmute yourself and then ask the question. Mr. Muhammad doesn't seem to be here. Okay, Manuel David, are you ready with the question from Nigeria? Mr. Emmanuel David, ask your question, please. There is so much noise here. Can't hear you, Mr. Emmanuel David. There's so much noise. Maybe Dr. Farouk, you can ask your question. We can't hear anything. Sorry? Mr. Muhammad, Dr. Muhammad, or Dr. Farouk, you can ask your question, then we conclude this session. Muhammad Farouk from Egypt. We can't hear anything on our end. Okay, it seems people are not ready for the question, so maybe we wait from AARDO to guide us into the valedictory session. Mr. Sanjeev Behera.
Hello, you hear me?
Yes, please take over, yes.
Yes, thank you for your nice presentation. I want to ask about 2 questions. The first one of weeds. What about the using of micronic sulfur to control weeds? The second questions of nematodes. What is the biological control can be affected to control the nematode of the larvae and the eggs, or one of them. I want to have one comment also about the control of weeding. I totally agree with Doctor about the certified and accredited seeds. It is very necessary to use to control weed, and also the good agriculture practice for preparation the soil and adding the good composting free of weeds and nematode. It is I agree for that, and also I want to comment also about the control of nematode. The control of nematode must be the farmers using the good composting, not using the fresh manure to the soil without any composting, because it is the source of nematodes and of the weeds. Thank you.
Thank you very much, Dr. Farouk, for those. Thank you for those questions and comments. Indeed, your comments are very, very pertinent towards dealing with nematodes and also weeds. Now to your questions. I am not familiar with the mycronic sulfur, so I will not answer that one. But the biological control to deal with nematodes, you are asking about targeting larvae and eggs. So it's better to, so a lot of the biological control options that we have for nematodes, they're actually targeting the actual nematode itself. As you know, nematodes do produce, they do lay eggs. Of course, they are not, they go from eggs, they go through, through juvenile stages. You have the juvenile 1, juvenile 2, which they're just like snake-like things. They're just growing in size as they are molting. Okay, so that is the stage that is typically targeted by a lot of the biological control options that are available. But I agree with you. It would be nice to expand on that and find something that can also attack the eggs, especially for nematodes like cyst nematodes, where you have these eggs, protected by this cyst. Yes, but most of what we have, the microbes we are using now, they are targeting the actual nematode, the actual juvenile or the actual adult. Thank you, moderator. Over to you.
Okay, thank you, Dr. Paul, for answering those questions. Now, because of time, we won't continue with the question. We have to transition into the valedictory session. So thank you very much for participating in this session. So please, you can just continue leaving your comments with maybe in the chat box. We'll get back to you. The presenters will always get back to you somehow, maybe through email or something. So thank you. Unfortunately, we have to go into the next session because of time. So just give maybe 30 seconds and then we start our valedictory session. Thank you everyone who participated in this. Dr. Paul, you can close your presentation, then we go into the next session. Thank you.
Thank you very much for all. I Thank you very much for all. I have another meeting. I leave now. Thank you. Bye.
Thanks to all. Goodbye.
So good afternoon, everyone. So we'll start our
Thank you.
session. Yes. Good afternoon. So the last session now is the valedictory session. Hello. So the valedictory session has started. We've got a program which is just about 30 minutes to the most. So I hope the people from AARDO are present. We'll start with the presenter, the one who was presenting last, Dr. Paul, just to the head of Department for Plant Science at the University of Zambia, just to give us just a briefing on how the training was and the program in terms of organization of the training and his experiences, and just a few words about the training, how it went. We'll just give him a few minutes to say something about the training and the insights about it. So Dr. Paul, over to you, please.
Thank you very much, moderator, and thank you for this opportunity to say a few words. We are very happy, very glad as an institution for how this training has gone. In this training, we were able to cover 4 major areas, that is modern concepts in insect pest management. We looked at the role of plant breeding in plant disease and pest management. We looked at modern concept in plant disease management, and today we closed with the session on weeds and nematodes. Of course, these are not exhaustive, going by the nature of the topic. As you will agree with me, even from the question and answer sessions, it used to go on and we just have to cut it. That shows the needs in those areas. It was really an important training, but clearly there was need for more, but we are very happy because it has covered the basics. We had more than about 140 people on each of the days, so we were very happy with this attendance. And of course, there were more who were watching us through live stream via Facebook and other media. I don't know what the numbers are there, but all in all, we are very excited as UNZA, and we are very happy with how this training has gone. In terms of our experiences in preparing this, I would like to really say thank you to AARDO for the way our collaboration with them has played out in this. It's without their dedication, it would have been difficult to pull out this training, but they've been very instrumental. We are very thankful to the collaboration with them, and we look forward to more. Of course, as an institution, we are very young in terms of the interaction with AARDO. This was our first time, but we are very positive going by how things have gone. For example, when we were in the process of preparing for this training, the interactions that were going on between our contact person from here, Dr. Mfungwe, and Mr. Kamau from AARDO. Mr. Kamau was very timely with his responses and very precise with guidance as we were consulting in the process of doing this. So, Mr. Kamau, thank you very much. You made preparation of this training very pleasant. Okay. Yeah. Then
Thank you, Professor.
yes, thank you very much. Also, in terms of future, we are looking forward to future collaborations because the topic we tackled today, I mean, during this training, is just one of the areas in which the institution has expertise in, but also in which the institution is interested in collaborating with other researchers. To start with, on your part, AARDO, we do have other departments, I'm sure you are aware. Even within this department, plant science, which was handling this training, we still have many things to do with horticulture, for example, several other fields to do with physiology and so on that we could still get into and participate in some training in the future. As a school, also, there's several other departments. There's departments that deal with things on the animal production side, departments that handle with food science and nutrition, agri-economics, soil science, and so on. So really room for collaboration and future training is vast, and we are very open to you, AARDO, and we are very, very thankful for how this preparation has gone through and for the potential collaborations that are still upcoming. To the participants, we are very thankful for your overwhelming response. I have seen names that have consistently showed up from the first day we started this up to today. I've been looking at the register. Every day they are coming and every day they are asking questions. So we are very, very thankful for that interest that has come from you participants. We know that you as participants, you also represent different institutions that do training in many areas, that do research in many areas. Please reach out to us, and I hope you'll be open. We'll try to reach out to you. Let us collaborate more. We are very, very thankful. We know you have lots of experiences in several other areas where we as researchers from UNZA can benefit from. So we look forward to interactions with you. And of course, we are looking forward also to your comments on how this training has gone. I know Mr. Kamau maybe has a formal way of collecting this feedback from the participants. But if any of you participants want to send an email to any of us here at UNZA to give us your feedback, we would be more than happy to. Thank you very much. Over to you, moderator.
Okay, thank you very much, Dr. Paul, for the kind words. Indeed, we have really benefited a lot from the interaction, even as we were hosting this training. So thank you for those words, indeed. So now we want to get some feedback from you, the participants. So in this session, we just want to hear what your reaction has been towards this training. How has it benefited you? What is at least 4 people, maybe from Asia, 2 from Asian countries, 2 from African countries, and just say something about the training, because we've just recently joined the AARDO group. We want also to learn something from you, in areas where we need improvement. What has been your experience in this training? Can I have some hands, maybe where I can choose people to give us feedback? Who is going to volunteer to just give us a few words, the feedback from you, the participants? You are the ones who are getting the training. What is your experience during this training? I can see Mr. Richard Diego from
Hello?
Ghana. Is your hand up?
Hello?
Yes, Mr. Richard. You can say something?
Yeah, I want to say something.
Okay.
Yeah,
Go ahead.
thank you, Madam Moderator, and thank you, Dr. Paul. I think AARDO and collaboration with University of Zambia have done effective training for us, the participants. Honestly, the 4 days training have been much in-depth in terms of knowledge. I've acquired a lot, and I wish AARDO will collaborate more for us to join more trainings and advance our knowledge in terms of agriculture to increase food security in the world and Africa in particular. Thank you.
Thank you for the wonderful words, Mr. Richard. We really appreciate.
Thank
Dr.
you.
Farouk, you can also go ahead.
Hello.
And, yes, go ahead.
Hello. Thank you for your organized, and thank you for Dr. Paul, and thank you for all participants. According to my position as associate professor biological control of plant pathology, this course is very important for me because this course is search about the new technology don't harmful the environment and the plants or the human. So during the training course, I acquired many concepts about the controlling disease without any chemical disease, because this is more safety for the human and don't harmful the environment and keep the environment is clean. Also, I am admired with the administration of the soil, because almost adding the composting, which enhances the soil with organic matter and fertility. But I want this course is applied at Zambia, because the practice and so the experience from the on the field, it is more effective, and it is more high experience for at this field. Thank you for all, and thank you for this training course and for your all organizer in this course. Thank you.
Thank you very much, Dr. Farouk, for your kind words. We really appreciate your feedback. Maybe allow also Ms. Aisha Dambo Gambia to just say a few words. Aisha, you can go ahead.
Yes, good morning to you all. My colleagues have said it all, but I would like to add a few words, please. The level of experience displayed in this platform is phenomenal. I really appreciate all of you, the University of Zambia, as well as AARDO, for this phenomenal job. It was experience. I have been attending trainings with AARDO in collaboration with others, other institutions, but like this is one of the best. Yeah, the experience is just beautiful. The knowledge display is phenomenal, and I really appreciate that. And the topics are also very important. But we would also appreciate if there will be future collaborations. That would be beneficial to me and of course, my colleagues, I believe. So thank you all for this job. Thank you for working hard. We know that there are many who are involved in this great job, because there are some behind the screen, but at the end of the day, you all participated well, you all did well, you worked together to have such a phenomenal training, to make sure that we get all what we need during this training. And I thank you and applaud you for that. Thank you all. And thank you to my colleagues for being phenomenal people with your very good questions. I learned a lot actually from you all. I thank you all for being there, and I thank Allah, and I applaud them as well, because like this is maybe about the 11th training I've been taking with Allah. So, and I am really grateful for having this opportunity. Thank you all.
Thank you very much, Ms. Aisha, for those kind words. Thank you for encouraging us. We can also get a word from Dr. Sabina. So Dr. Sabina Fatima from Pakistan.
Yes, I am Dr. Sabine Fatima from Pakistan. It's a very fruitful training regarding modern concepts in plant protection. And it is suggested from my side to please open new ways for the professionals to physically visit the places for the exchange of experiences like Zambia. Thank you.
Thank you very much, Dr. Sabine. Very short and to the point. Thank you very much. Shama Abdu from, is it Sudan? You can say a few words. Shama.
Hello.
Yes, go ahead.
Hello. I would like thank you to University of Zambia and AARDO, and all thanks to the trainer and trainees. I am happy to participate and benefit from the training, and I wish the best for everyone. Bye. Goodbye. Thank you.
Thank you very much for the kind words. Is it Adiola Abo from Adiola? You can give your words.
Thank you so much. I want to appreciate AARDO and University of Zambia for this online training. It's been superb. The organization has been superb. Then for time management as well, I really want to appreciate you for the in-depth knowledge shared. I appreciate the facilitators, the names, all the facilitators, I appreciate them. And also for the clarity in presentation, well spelled out, well thought. I appreciate every one of you. Then I also want to appreciate our coordinator, Dr. Joyce, for the job well done, and Mr. Kamal. Thank you.
Thank you very much. I'm seeing some hands still going up. We won't accommodate everyone, but maybe the one who's been up for a long time. Josiah Kamati from Namibia, you can go ahead. Josiah Kamati.
Thank you very much. So thank you very much. I really appreciate the opportunity. So, and we learn a lot. I learned a lot, in particular from the Namibian concept. As we know, Namibia is one of a developing country in Africa, and food productivity is not really good. And with this information, it will help a lot. And I appreciate everything, and I'll make sure that whatever I acquire will be implemented, and it will make a significant change in the Namibian people. I thank you very much. Thank you very much.
Thank you very much, Tim, for all those kind words. In the interest of time, I think we'll end here with the feedback from the participants. Maybe we'll go to the next item. We'll get a valedictory address from AARDO, and Dr. Sanjeev Behera, the head of IEC division of AARDO, will give the valedictory address. Dr. Sanjeev, you can go ahead with the address, please.
Thank you very much, madam. Am I audible?
Yes, you're audible. You can go ahead.
Okay, thank you. Thank you very much. Dear Dr. Paul Kachu Pululu, I'm sorry for the pronunciation, head of the Department of Plant Science, School of Agriculture, University of Zambia, and the principal course coordinator of this online training program, Madam Dr. Joishi, co-course coordinator from the School of Agriculture, University of Zambia, my colleague Mr. Kamal Dhameja, technical officer, other officials presented from AARDO, and dear participants. My team, along with the learned faculty members of the University of Zambia, while planning for this online training program, they set a set of objectives for this program. And those objectives were: identify the challenges associated with traditional methods of plant disease and pest management, to recommend affordable, adaptable modern technology and mitigation solutions for pest and plant diseases to reduce crop losses, and come up with a model of an ecosystem that best strategy that focus on long-term prevention of pest of their damage through a combination of technology. When these objectives were brought before me, I was not very sure as to how this online training program will address these objectives. More precisely, the third objective, which talk of a model on an ecosystem-based strategy that focuses on long-term prevention of pests or their damage through a combination of technology. However, after listening to learner speakers during the deliberations and meaningful interaction by the participants, I am personally now satisfied that justice was given to the objectives despite the limitation of a virtual training program. I must congratulate all resource persons and Dr. Paul in particular and his team for completing this training program so successfully. As you all know, things are improving in the past few months and we are coming out from the COVID-19 pandemic, which is not turned out to be an endemic. And the whole world has started to come back to normalcy. We at AARDO have also started gradually opening up for the offline training program. I remember Madam Dr. Shabina from Pakistan was referring to the offline training program. And I wish very shortly we will have some offline training program also in the University of Zambia, which is spread over a vast area within the capital city of Lusaka and has a beautiful landscape with a water body right in the middle. In fact, I visited this university way back in 2010 and stayed there for 2 days. And really, when we'll go for an offline program in the campus itself, the participants will learn more and better, definitely. Dear participants, as I mentioned, AARDO has already started organizing offline training programs. I request all participants to keep an eye on the AARDO website. However, the number of participants would be much less and maybe compared to 10-15 for a program like this at best. And we accept nomination through our nodal ministry only. So I suggest that for offline program, please apply through the nodal ministry of your countries. However, with the online programs, when we evaluate the success, we have decided to continue with the online program as well. Besides short-term course, as like this one, we have signed several MOUs with several centers of excellence across Asia and Africa for master and PhD program in the field of agriculture and rural development. Many young student officers from AARDO member countries are now pursuing their higher studies in different IITs, that is for Indian Institute of Technology in India and other institutions in Korea as well.
I'm sorry, Dr. Jose, I think some technical glitch is there. Dr. Bahar is reconnecting. Just give a second. Huh?
Oh, okay, we'll give you a second. Yeah, he is dropped off. Hello?
Yes, Doctor.
Okay, we'll wait a few seconds, though.
Yeah, there, he's going to just rejoin it.
Okay, we'll do.
Yes, Dr. Bahar is with us.
Oh, okay. You can go ahead, Dr. Sanjeev. You can continue with your presentation.
Sir, you're mute. You have to unmute first.
You're muted. We can't hear you.
Okay. Am I audible now? Hello?
Yes, you're audible. You can go ahead.
Okay.
Thank you.
So actually, I was talking about the online program actually. So as I said that actually we will continue with the online program even if we start the offline program. And I was referring to the MOU that we have signed with various leading institutions across Asia and Africa, including Indian Institute of Technologies, and some of our students from AARDO member country. They are now actually pursuing their higher education in different IITs in India and also some institutions in Korea. We are in touch with several other universities across Asia and Africa, including the host institute, that is the University of Zambia, for some fellowship to the students and officials of AARDO member countries. It will expose AARDO's target population to acquire new scientific knowledge and bring new knowledge and technology to their respective country. As you are aware that AARDO stands for South-South cooperation in the field of agricultural and rural development, we are focusing more now on the transfer of technology, transfer of knowledge, technology which are affordable and adaptable among the member countries. If you go to our website, you will find a basket of such technology parked there under the heading of ATM, Affordable Technological Menu. Recently, in a function held in New Delhi, 5 such affordable technologies were handed over to 4 AARDO member countries. We encourage you to send us any innovation in our areas of concern, that is agriculture and rural development, so that the same can be parked for the larger benefit of the people of both Asia and Africa. Ladies and gentlemen, I don't want to take much of your time. On behalf of our Secretary General, His Excellency Dr. Manoj Nardev Singh, and our Assistant Secretary General, and on my own behalf, I extend my sincere thanks to all involved in organizing this online training program from both University of Zambia and AARDO, and also to the country government who spare their officers for these days, and dear participants for your active participation. Thank you very much. Thank you for your patience learning. Long live South-South cooperation. Thank you.
Thank you very much, Dr. Sanjeev, for that address. We really appreciate for encouraging us. So now we come to almost the end of the program. I'm going to give a vote of thanks. So in my vote of thanks, thank you for giving me this privilege also to do my vote of thanks. So first thing, I also greet everyone who is participating in this valedictory session. I just want to thank you everyone, and a special thanks also to His Excellency Dr. Manev, the Secretary Manov, the Secretary General of AARDO, and the whole team at AARDO for also sponsoring this training workshop. We really appreciate. And also special gratitude go to Dr. Kamau Dame for supporting and guiding us throughout the preparation period. Dr. Kamau was very generous also with his time, and we were able to consult him from time to time on how to go about this training program. We really appreciate the time that you spent with us in preparing this workshop. And also I give special thanks to the trainers at University of Zambia, Dr. Paul and the whole team, who is the head of department, the whole team. They spent time also putting in a lot of time in preparing and also delivering the presentations, which people have enjoyed. Thank you very much for the time that you spent in this. Also special thanks also go to the management team at the University of Zambia, and specifically the School of Agricultural Sciences, for allowing us to represent them in this training program. We really appreciate them. And also we can't also without appreciating you, the participants. Thank you very much for making this training session very memorable. Your comments, your questions, and your high level of engagement is really appreciated. We really appreciate your engagement. This is the first time we are presenting at an international forum, so we appreciate your cooperation and your kind words and your support during this training program. Thank you very much. There is a saying that iron sharpens iron, and we know that we are sharpening each other. Since we've joined AARDO network, we hope to learn a lot from each other going forward through trainings, technology transfer, and also through exchange of good practices and many more, just like Dr. Sanjeev was mentioned. This is not the end of the training program. We'll be collaborating. This should open actually networks where we can network on an individual level and also at international level through our institutions. So this, I'm sure, will open a lot of doors for us and for everyone else. We appreciate the cooperation. Also, I thank you for each one of us for attending this workshop. I cannot say thank you enough for you people. Thank you, thank you. It has been a very good 4 days where we've interacted with you, where we continue to look forward to more interactions in future. Thank you very much. I end my vote of thanks here. Thank you very much.
Thanks a lot.
Yes, Dr. Kamau, probably you can guide us in our next issue.
Yeah, thank you, Dr. Jose, and thank you, Sanjeev sir. Thank you, Dr. Paul. The only that at the end, I just say that it was a really amazing group. That one session was going on here on the online platform, and second was going on the WhatsApp group. So there was parallel two discussion were going on for this high level scientific participants. We got it. The only submission is with regard to the certificate. We are working on it, so it will take 7 to 8 days. It will be dropped on your email. And also I request you, I have already shared feedback form. So most of the feedback form I have received. I have also sent the link on your emails also. So you are kindly requested to fill your whatever the feedback is. And as I have seen in the chat, you have mentioned about the offline program. You can also mention in the feedback form. So that will also allow us to prepare our roadmap for the upcoming training programs. Thank you very much once again. Thank you. So now we can leave the meeting, Dr. Jose. So
Yes. Maybe I can just mention a few words. I'll post also on the fora the attendance list for the people we think have attended the session, at least 75% of the sessions, so that we agree on the number and also the names. Make sure you correct your names the way you want them to appear on the certificate. You have to correct that, and also on the number, the attendance, we agree. I'll put the final list, and then we can start from there. Thank you.
The only is, Dr. Jis, I wanted to just add it because some of the participants having an issue with the internet or all this, so they are following on the YouTube live link. And so therefore that we have mentioned one section in the feedback form, they can confirm how many sessions they have attended. So accordingly, we'll work on the feedback for the certificate according to their feedback. Thank you, Dr. Jose.
Okay, thank you very much.
So
thank you.
Thank you very much.
Okay, thank you.
Thank you very much. Thank you very much, and see you shortly. Bye.
Thank you. Goodbye.
Thank you.
Thank you, and goodbye. Please.
Bye.
Thank you.
Goodbye.
Thank you very much.
Please
Thank you.
for an
Goodbye.
hour.
Thank you and goodbye.
Thank you so much. Goodbye.
Thank you. Thank you.
Thank you, and goodbye.
Thank you. Goodbye. It's been a very good time. Let's that time. Bye. Bye.
Thank you. Goodbye.
Goodbye.
Bye.
Goodbye.
Thank you.
Goodbye. Thank you, and
Thank you
goodbye.
very much. Bye-bye.
Thank you. Goodbye from Ghana.
Bye.
Thank you, Ardo. Thank you. Bye.
Thank you.
Thank you.
Goodbye.
Thank you. Thank you. Thank you. Bye-bye.
Thank you, and goodbye.
Thank you.